Hello everyone, this is educator Priyanka Thakur teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. So today in this video I'm going to teach you about bacterial taxonomy and bacterial toxins. Okay, so let's start with our today's topic. When we talk about taxonomy, we talk about what? We talk about identification, nomenclature and classification. So in this process of classification, organisms are arranged into groups. They are organized into species, genera, families, and different higher categories. There are main 7 obedient categories. So the basic taxonomic unit is species, and each species is organized to form a genera. The definition of eukaryotic species is different, while prokaryotic is different. For eukaryotes, the definition of species usually stresses on the ability of similar organism to reproduce sexually with the formation of zygote and produce fertile offspring. Okay, it means a species is a group of individuals which are having similar characteristics which can interbreed and can produce fertile offspring. But bacteria do not undergo sexual reproduction, so this definition of species does not apply on bacteria. Okay, we cannot apply this definition on bacteria also. Okay, so what exactly is bacterial species? Bacterial species is a group of organisms that will share many phenotypic properties and it will have a common evolutionary history. More recently, if you're talking about bacteria, more recently we talk about, so bacteriologists define bacterial species based upon genomic similarities between strains. According to these two strains, if of same species, must have very similar composition of very similar mole percent of guanine cytosine content, and this percent is specific but not exclusive. Like for a species, two strains with similar guanine cytosine content may or may not belong to same species. But if two strain are very different, like their guanine cytosine mole percent is very different, then they must not belong to the same species. What exactly is a strain? So a strain is the category that is found below the level of species, and we can say that it is just like that of subspecies. Okay, and the strain consists of descendants of a single pure microbial culture, right? Okay, and remember, the more is a DNA is more DNA is related, higher will be its reassociation ability. Remember this thing. And DNA relatedness by the method of DNA reassociation is determined by how you just allow single-stranded DNA from one strain to reassociate with single-stranded DNA from a second strain. It will form a double-stranded DNA molecule, okay? And this is specific temperature-dependent reaction. And the optimal temperature for this reassociation is approximately 25 to 30 degrees Celsius or below than this. Okay, and at this temperature, native double-stranded DNA can denature or renature. Fine. So strands can denature or renature. You understand it? But higher the ability of two strains to renature, to reassociate, more is their common guanine, cytosine content, right? Okay. So a category, I told you about the category which is found below the level of species in bacteria. This is strain, and what is strain? You understand about strain now. Classification. So there are different classification system. One is phenetic, other one is phylogenetic. When we say phenetic classification system, what exactly is the classification system? Organisms are grouped together based on their overall similarity to form a phenetic system, or they are grouped based on their evolutionary relationship and it form a phylogenetic system. Let's talk about phenetic system first. So what is a phenetic system? It is a system of classification that will group organisms together but based upon their mutual phenotypic similarities. And this term phenetic was coined by Kane and Harrison, and it means a relationship by overall similarity based on all available character without any weighing. Phenotypic features are compared without assuming that any feature are more phylogenetically important than others. Although the phonetic system that it can reveal possible evolutionary relationship, but it is not dependent upon phylogenetic analysis. On the other hand, phylogenetic system most of the bacteriologists follow this phylogenetic system of classification for bacteria. This system also consider evolutionary relationship, and one major constraint, when we talk about constraint, we I hope you understand the meaning of constraint, like something that is not allowing, okay, us to do a particular thing, okay. So one major constraint in the development of phylogenetic system is what? Bacteria do not have a fossil record, but it was overcome by comparison of genetic material and gene product, okay. So comparison of genetic material and gene product have overcome these problems. Total DNA of one bacterium can be compared with that of any other bacterium, and five factors can be used to determine DNA relatedness, like your guanine cytosine content, okay, DNA relatedness under condition optimal for DNA reassociation, thermal stability of related DNA sequences, genome size, DNA relatedness under conditions supra-optimal for DNA reassociation. Okay, so all these are the five factors that will be uh, telling us about what determining the DNA relatedness. Okay, now let's talk about some general feature of important bacterial group. Let's first talk about Rickettsias and Chlamydia. Rickettsias are gram-negative bacteria. They are mostly non-motile and obligate intracellular parasite. It means they will require a host, and they are named after their discoverer. His name was Taylor Ricketts. And these species are parasite on vertebrates, and their vectors are like fleas, ticks, lice, and mites. They are causative agent of several human diseases like typhus fever, Rocky Mountains. Okay, they are causative agent. Okay, they themselves the causative agent. It's not like they are causative agent. Okay, they are causative agent for typhus fever, like R type 5 for typhus fever, Q fever, spotted fever. Okay, and then chlamydia. Chlamydia is just like Rickettsias. It means they are also obligate. That's why I have not written here. They're also obligate intracellular non-motile gram-negative bacteria. And in this, they have one class, one order, four families, six genera, and among six, your chlamydia is best studied. These cells are unable to carry out energy metabolism, and they lack biosynthetic pathway. Therefore, they are entirely dependent on their host to supply them with ATP and other intermediates. They are dependent on host. That's why they were also considered to be viruses, but they are not viruses because they have cell wall, DNA, RNA, and ribosome. They reproduce in the cytoplasmic vacuole of their host, and they undergo two stages, like the infectious particle, which is also known as elementary body, and intracytoplasmic reproductive form, which is also known as reticulate bodies. When we talk about elementary body, its size is smaller as compared to reticulate body, and it is more infectious than reticulate body. Elementary body is usually inactive, and reticulate is usually active. And elementary body survive in extracellular space, while reticulate body can survive is adapted to survive in intracellular subspace, and trypsin digestion is resisted by elementary body, while reticulate body is sensitive for trypsin digestion. Okay, these are some of the comparison. These are comparison between bacteria, rickettsias, chlamydia, and viruses. So see, intracellular parasite bacteria are not truly typical, like they are not intracellular parasites. Okay, rickettsias are yes, and chlamydias are also intracellular parasites, viruses as well. Binary fusion occur only in bacteria, Rickettsias, chlamydias, but not in viruses. DNA and RNA both are found in bacteria, Rickettsias, chlamydias, but virus either contain DNA or RNA. Ribosomes. Bacteria, Ricketsia, Chlamydia both have ribosomes. Virus does not have ribosomes. And bacteria are sensitive to antibiotics. And Ricketsia, Chlamydia are as well sensitive to antibiotics, but viruses are not sensitive to antibiotics. Sensitive to interferons. Typical bacteria does not have any sensitivity for interferon, while Rickettsias and Chlamydia they also don't have any sensitivity to interferon as well, but viruses have the sensitivity to interferons. Okay, the next group comes to be Actinomycetes. These are aerobic, gram positive, and they are more like bacteria. They actually form a septate hyphae, and they have some asexual spores, just like that of fungi. And their asexual spores are generally coniospores and sporangiospores. And its classification is mainly based on properties of conidia, arrangement, presence of absence of sporangium, cell wall type, and they are mostly non-motile. And they are whenever there is motility present in them, they are called as flagellated spores. They have flagellated spores at that time. So, Actinomycetes, the largest known species we have studied is Streptomyces. So members of these genus are strict Arabic, and they are non-pathogenic, and they bear strain of non-motile conidia. Their natural habitat is soil, and the odor found in the soil is largely due to the production of volatile substances such as geosmin from the Streptomycetes. And it's Streptomyces, not Streptom. Okay, Streptomycetes are the best known photosynthesis of a vast array of antibiotics like chloramphenicol, erythromycin, neomycin, streptomycin, and tetracycline. Okay. Next comes to be spirochetes. Spirochetes are a group of gram-negative bacteria. These are chemoheterotropic bacteria. They are long, flexible, helical-shaped organisms. They do not have any external rotating flagella. They are creeping or crawling like of organisms, and this pattern of motility is due to their unusual morphological structure called axial filament. And the central protoplasmic cylinder, which contains cytoplasm and the nucleoid, is bounded by plasma membrane and gram negative type of cell wall. So spirochetes are they mainly represent gram negative. Okay, so there are two or more than 100 prokaryotic flagella called axial filament or periplasmic flagella. They extend from both ends of the cylinder. They often can overlap. Okay, so Treponema pallidum, which causes syphilis, Borrelia, which is responsible for Lyme disease, are some of the example of spirochetes. Okay, next come Mycoplasma and Cyanobacteria. You already have idea about Mycoplasma and Cyanobacteria. That's why I have not included these two topic here, like Mycoplasma and Cyanobacteria. You already know that Mycoplasma are also called pneumonia, pleuropneumonia like organisms, because they were first isolated from the cattle which who was suffering from pleuropneumonia. Okay, so the trivial term molecules is also used to describe any member of mycoplasma. Okay, and okay, and next is your like cyanobacteria. You also have idea about cyanobacteria, and they are cyanobacteria are actually gram negative bacteria. Remember this thing, cyanobacteria are actually gram negative bacteria. Fine, they are having the pigments, they are having the trichomes, homogonia, kinases. I hope you are aware of these terms. What are akinites? Akinites are the resting spores which help the blue-green algae to protect themselves during the unfavorable conditions. They do not have an enzyme alpha ketoglutarate dehydrogenase, thus they do not have a fully functional citric acid cycle as well. Okay, now let's talk about archaebacteria. Okay, bacteria eukaryotes, we have studied about them, and let's talk about archaebacteria. So archaebacteria, if we talk about archaebacterial morphology, reproduction, physiology, and ecology, it's quite like very diverse, and they can be gram positive, negative, and of any shape. Okay, they can be aerobic, anaerobic, or facultative or obligate anaerobic as well. They are very similar to your eubacteria, and they have similarity between archaebacteria and bacteria. Okay, they do not have nucleus, but they have histone proteins. Okay, they have spliceosome introns. Okay, their circular organization of their genome, organization of gene into operone, presence of 78 ribosome. All these are the characters which are similar to that of bacteria. Okay, so let's know about the differences between eubacteria and archaebacteria when we talk about nucleus. Nucleus is present in only eukaryotes. Okay, it's absent in bacteria and archaebacteria. No membrane bound organelles are present in both of them. Cell wall in bacteria is made up of peptidoglycan, which contain muramic acid, and archaea have variety of types of cell wall, but no muramic acids are found in the archaebacterial cell wall, and membrane lipids have ester-linked straight chain fatty acids in bacteria, while archaea does not have any ether-linked branch aliphatic chains. They have ether, not ester. They have ether-linked branch aliphatic chains. Transfer RNA in transfer RNA, thymine is present, and no thymine in transfer RNA is found. And N-formal methionine carried by initiator tRNA, while methionine carried by initiator N-formal methionine carried by initiator tRNA, while methionine is carried by initiator tRNA. Okay. So ribosomes, elongation factor 2 it does not react with diphtheria toxin. Okay. So in a like bacteria, the elongation factor 2 does not react with diphtheria toxin. But in archaea, it does. Sensitivity to chloramphenicol, bacteria are sensitive, archaea insensitive, while bacteria are insensitive for anisomycin and archaea are sensitive for anisomycin. Then come DNA dependent DNA polymerases. Okay, there are one number DNA dependent DNA polymerases. In archaea, there are several. Bacteria are sensitive to rifampicin and archaea are insensitive to rifampicin. And the bacteria does not have polymerase 2 type of promoters, but they are present in archaea. And they in bacteria, you won't see any methanogenesis. Archaebacteria, and they are responsible for methanogenesis and chlorophyll based photosynthesis found in bacteria, but it is absent in archaebacteria. Okay, so all these things are clear to you now. Other than this, are the you know that there are major group of archaebacteria like methanogens, halophile, thrombophiles. I have already discussed all these things in my previous video. That's why I'm not going to talk about this thing here. Now let's talk about the bacterial toxin. So toxin is a specific substance, which is often a metabolic product of organism that damages the host. It can induce the disease in the absence of organism that produce them actually. Okay. So when disease results from the entrance of a specific toxin into the body of host, it is intoxication. And when toxin enter the blood of host and disease is caused, this is known as toxemia. There are mainly exotoxin and endotoxin. So what is the source of exotoxin? Certain species of some gram-positive and gram-negative bacteria are the source of exotoxin, and endotoxin are mostly produced by gram-negative bacteria. Exotoxins are secreted by cells, while endotoxins are not secreted by cells. The exotoxins are polypeptide, endotoxins are lipopolysaccharides. Exotoxins are found in plasmid or bacteriophage, and endotoxins are found in bacterial chromosome. Toxicity is high for exotoxin, low for endotoxin. Antigenicity induces high titer antibodies called antitoxins because their toxicity is high. That's why they must have a high antigenicity, while endotoxin have poor antigenic properties. Okay, so vaccines can be produced from exotoxins. So these are toxoids. The term given to such vaccines is toxoids used as vaccines, and no toxoid formed and no vaccines available. And they are exotoxins are destroyed on 60 degrees Celsius, while endotoxins are still stable on 100 degree centigrade for 1 hour. Okay. So types of exotoxin, there are neurotoxin, enterotoxin, cytotoxin. Okay, that's very obvious. Like exotoxin, they are divided on the structure and physiological activities. When they are exotoxins are also categorized on the basis of site affected, like neurotoxins, nerve tissue, and neurotoxin, intestinal mucosa, cytotoxin, general tissue. Okay, then toxin that also act as on the cell surface. So toxin that act from the cell surface bind to a receptor on the cell surface, and that will stimulate intracellular signaling pathways. One common example is supraantigens. Supraantigens are produced by several bacteria. The best characterized supraantigens are produced by Staphylococcus aureus, Staphylococcus pyogenes. They cause toxic shock syndrome. Actually, supraantigen will bridge or measure histocompatibility complex 2 proteins on antigen presenting cells, and with they bridge a measure histocompatibility class 2 protein on antigen presenting cells with the T cell receptor on the surface of T cells. Okay, on the surface of T cells. So ultimately 30% of T cells are activated, and they are leading to very massive secretion of cytokinin, which produces symptoms of toxic shock. There are also membrane damaging toxins which will form channel forming. These membrane damaging toxin will disrupt the integrity of plasma membrane. Okay, so they are channel forming toxins, and they are toxins that enzymatically damage the membrane. Other are AB toxin. If I talk about AB toxins, okay, so AB toxins are actually intracellular toxins. The B subunit will attach to target region, and A will be acting as an enzyme. So if we are using only A, it will be it can cause enzymatic activity. It is enzymatically active, but it lacks binding and cell and entry capability. Whereas isolated B subunit bind to target cell but are not non-toxic and biological activity. Like cholera toxin, pertussis toxin, your tetanus toxin, all are AB toxin. So these are the bacteria and diseases and how they perform their action. Corynebacteria diphtheriae, gram-positive bacterium, cause diphtheria, inactivated elongation factor 2 by ADP ribosylation. Very important chart it is. You should remember this chart. Clostridium tetani causes tetanus. How? It blocks the release of inhibitory neurotransmitter glycine. Clostridium botulinicum causes botulism. It blocks the release of acetylcholine. Clostridium perfringens gas gangrene causes by alpha toxin. Alpha toxin is a lecithinase. Actually, Clostridium perfringens is again your alpha toxin, which will act as lecithinase and will disrupt the membrane. Bacillus anthracis causes anthrax, which is one of the toxin, and it acts as an adenylate cyclase. Okay. Staphylococcus aureus produces toxic shock. It binds to the class 2 major histocompatibility proteins and induces interleukin 1 and interleukins 2. Streptococcus causes scarlet fever, and the mode of action is unknown. Gram-negative bacteria include E. coli, Vibrio cholerae, Bordetella pertussis. E. coli causes watery diarrhea and bloody diarrhea. When it causes watery diarrhea, they are labile toxins which will stimulate adenylate cyclase by ADP ribosylation, and stable toxins stimulate guanylate cyclase. While in bloody diarrhea, virotoxin is cytotoxic to enterocytes. They release virotoxin. E. coli releases virotoxin, which is cytotoxic to enterocytes. And Vibrio cholerae causes cholera. It stimulates the adenylate cyclase by ADP ribosylation of GS alpha, while Bordetella causes whooping cough by the same action, but it causes the ADP ribosylation of GI alpha. That's all for today. If you are preparing for CSIR net, you can join an academy and use the referral code Neha. And you can follow, this is the referral code of Neha Tanisha ma'am. You can follow her on Academy. And along with this, do not forget to subscribe to our channel, press the bell icon for joining us anywhere else. You can just check out the description box there. You will get the links to our different platform. On a single click, you will be there with us. That's all for today.